Okay, so today we're not only going to be talking about chemistry, we will talk about binding as well. And we should already be fairly comfortable with discussions of binding, since non-covalent interactions of these types of biomolecules we've covered in this class have been a point of focus up until now. We will be talking about 3 different serine proteases today and comparing them to learn how each of them binds different substrates. Let's start with the first one, chymotrypsin. So we know chymotrypsin binds bulky hydrophobic residues right before the cis bond, so right before the bond that's supposed to be cleaved. And on the right-hand side of my screen here, what you're seeing are images of 3 different binding sites the enzymes use. So if chymotrypsin is binding a bulky hydrophobic residue, what should the binding site look like? Well, it should be complementary, right, folks? And here we see that if we're binding something bulky, the binding site itself should probably allow for that bulky thing to fit. So we have small residues like glycine, like serine, that allow this phenylalanine to come in and form nice interactions with that binding site. You'll also see that the majority of amino acids pictured here are nonpolar, right? And so if we're binding a hydrophobic R group, we want the binding site to also be complementary or nonpolar. Of course, serine is a polar residue, but if I were to show you kind of 3-dimensionally what this binding site looks like, you would see that the vast majority of amino acids that comprise the binding site are nonpolar. So, chymotrypsin binds a bulky hydrophobic residue. The binding site has small, nonpolar residues. What about the next one, trypsin? So trypsin binds positively charged residues before the bond that's going to be cleaved. What does its binding site look like? Well, here we're binding lysine. Lysine is fairly long, so it's on the bigger side of amino acids. So you see that to accommodate its size, we have 2 glycines, right? The smallest possible amino acid. But interestingly, now we have a negatively charged amino acid. So this is definitely bringing in Coulomb's law here, folks. If we have a positively charged amino acid to bind, a negatively charged amino acid would form a very strong, favorable, attractive association. Finally, let's talk elastase. So elastase binds small and neutrally charged residues. So not nonpolar, but just neutrally charged. In the case of the picture, we are seeing an alanine being bound. It is small, and it certainly is neutral. So what do we see in the binding site? Where if we're binding something small, you see that the R groups in the binding site are actually a bit larger to help reach the R group that needs to be bound and kind of fit it more snugly. We have valine in there, which is nonpolar and neutrally charged, and we have a threonine in there because this enzyme, since it binds neutral residues, it probably also needs the ability to bind things that are polar but also neutral. So we have a polar but neutral amino acid in the binding site, as well as a nonpolar amino acid in this binding site, but both are slightly larger to help accommodate a smaller residue being bound. And you can think to yourself, folks, what if we needed to bind a large polar amino acid? what would the binding site look like? What if we needed to bind a small negatively charged amino acid? There's kind of all sorts of combinations that you could think through. And so before we leave for the day today, I want to show you kind of a holistic picture of the active site of serine proteases and one additional aspect of this active site that is important. So before we get into the slide, let me just orient us to what we're seeing here. So serine, histidine, and aspartate, right? That's our catalytic triad. That's our catalytic site. Down here, folks, in less detail, this is the binding site. Here we have chymotrypsin being shown. There is one additional location that I want to point out for us today, folks. It's called the oxyanion hole. And it is a location in the active site that stabilizes the oxyanion. And so let's talk through this. It's easier to see in the picture on the right-hand side. So on the right-hand side, you see we have our tetrahedral intermediate with a negatively charged carbonyl oxygen. This negatively charged carbonyl oxygen is the oxyanion. So how does this active site stabilize the oxyanion? You see that there is a hydrogen bonding interaction happening between the amino group of serine, as well as a hydrogen bonding interaction happening between the amino group of glycine and the negatively charged oxygen or oxyanion. These hydrogen bonding interactions help to stabilize this negative charge as it's happening in the reaction mechanism. We will see next time we're together additional examples of how enzymes can stabilize charge. Stabilizing charge in a mechanism is one of the kind of classic ways that enzymes behave as cofactors. So that wraps up things for us today, folks. We talked about the binding site and catalytic site of all enzymes within their active site. We saw the mechanism of RNase. We saw the mechanism of serine protease. We saw some acid-base catalysis and covalent catalysis as well. So I will leave you with our meme of the day, our only non-cat meme. Enjoy.